Hi everyone. In our last couple of videos, we've seen how a DNA sequence is used as a template to produce mRNA and the features of the genetic code. If you're not yet secure on these concepts, you may want to watch these first, as in this video, we'll see how the mRNA sequence is translated into an amino acid sequence to produce a polypeptide. We've already seen how mRNA is produced in the nucleus, and now that mRNA will attach to a ribosome, which is either free within the cytoplasm or on the surface of the rough endoplasmic reticulum. Before we look at the stages of translation, let's first examine the structure of a tRNA molecule. tRNA, or transfer RNA, is described as having a clover-leafed shape, and this is due to its base sequence allowing it to fold and complementary base pair with itself. At one end of the molecule, we find a 3-base sequence known as an anticodon. As we'll see a little later, this anticodon will complementary base pair with the codon sequence on the mRNA. Finally, it has an amino acid attachment site, allowing each tRNA to carry a specific amino acid to the ribosome. Translation begins with an mRNA molecule attaching to a ribosome between the large and small subunits. Next, a tRNA molecule binds to the mRNA codon via its complementary anticodon. The first tRNA to bind will be complementary to the start codon and carries the amino acid methionine. A second tRNA molecule now binds the next codon, bringing another specific amino acid. Condensation reaction forms a peptide bond between the amino acids. This requires energy from ATP. Once the peptide bond is formed, the first tRNA molecule is released as the ribosome moves along the mRNA to the next codon. The process continues until a stop codon is reached, and the completed polypeptide is released to undergo folding and further processing. And that's all for this series on genes and protein synthesis. Like and subscribe to receive more A-level biology content.